That's what do you call this? Net. And so this idea that the veins are running in all directions. So if you get that fixed in your mind, then these are going to look a lot different if you have a cross section. So imagine is you have a section cut through here, or you cut it through here. It's going to be a big difference because on the monocot, you're going to have a regular, orderly arrangement of the vascular tissue. You're going to have a cross section of every bundle. And over here, you might have a cross section, and then you go right to here, and you're going to be longitudinal for a while. And then you might get the cross again. And so this is about a structure and then visualization in 3D. So, would this be a dicot or monocot? Dicot. A reason?
You can't see
Here's
the
a
circle.
cross-sectional view of vascular bundle, and here's a longitudinal view. And I wouldn't get that if I was cutting across here. I would have nothing but cross-sectional view. Okay? Okay, now, here's one of the slides that you're looking at, and what I have marked here in brackets and arrows is some things you should know, such as, what would you call the single layer up here at the top of that bracket? That's the upper epidermis, and that makes this the lower. And then in the lower epidermis of this particular leaf, it's got some other structures. There's two cells here, one, two, guard cells, and they always occur in pairs. And here's another way you can recognize them. Is that, notice there's a little bit of space here. There's a stoma, and then here within the leaf, there's space. You can't have an opening and then have a cell right above it to plug it. That wouldn't make any sense. So you can quickly scan across the leaf and look, look, there must be a pair of guard cells there, but they're not as obvious as here because that's the stomata is closed. Then, what would be the collective name for all of this layer here? Mesophyll. Does it make sense why it's called meso and fill?
Yeah.
Because fill means no. That's P-H-I-L. P-H-Y-L-L happens to mean leaf. Okay, it's not love. Meso, middle. So this is telling you it's the middle part of a leaf. And we'll have several other terms in here with meso, and we'll have quite a number of terms with fill in it, P-H-Y-L-L. Okay, now what part of the mesophyll is this portion from there to there? Palisade, and this portion is spongy. Now some leaves have palisade and spongy. Some leaves have spongy only. I don't know of any leaves that have palisade only. Reasons for that, we're getting to. What is the name for this structure at the bracket from there to there? That's a vascular bundle. And then what is the name for the particular tissue at the pointer here within the vascular bundle? Xylem. You can tell even this magnification because of the color. It's as usual. It's red, quite different from the phloem, which is below it. And on all these leaves that we have today, the xylem, once you find it, you're going to look below for the phloem. Does anyone know why? It's just the way it is. No, there's actually a reason for it. And the best way to understand it is with a model over here. That this is to help you to visualize these things so you don't have to memorize them. It should make sense. That if you realize, we covered earlier, that within the stem, that if you find the xylem, you go to the outside to find the phloem. Now you have vascular tissue, and think of it as it curves into the leaf. So whatever was on the outside of the stem ends up on the lower surface of the leaf. Any other way would make it like twist. That makes it more complicated. That doesn't make any sense. So based upon the arrangement of the vascular tissue in the stem, the leaf goes accordingly. It should make sense. Now you might ask, well, why is the flow on the outside? We'll cover that next time. So by next time it is, you should have several answers to questions I've talked about. Like, why is the stele or vascular tissue of the root different from the stem? Why is the stem different from the root? Why is monocots different from dicots? Why is the vascular tissue in the stem the way it is? Hopefully after next lecture in lab, and certainly after we get into the chapter 5 lecture, we'll have answers to all those. And it should make sense under survival strategy. Okay, anything else on this slide? I think we've covered all my arrows. Okay, would this be a monocot or dicot?
Monocot.
That would be a monocot. There's a parallel venation, and so that means that this bracket here is a vascular bundle. And what do you call this layer here? Spongy mesophyll. There's no palisade. Why? We'll get to when we get to photosynthesis. There's a reason for this. Notice that this arrow here and the arrow up there, it's got stomata on both surfaces. So to help you with some of the questions you've been asking me, which leaf, this one or this one, would be more efficient and effective gas exchange, moving oxygen, carbon dioxide by diffusion?
The monocot.
The second one, right. And monocots or dicots can have this. So don't think all monocots have guard cells on upper and lower. It depends. But if you have them on both, you're going to get much more efficient gas exchange. However, there's some problem with that. It's good for photosynthesis, but what is it bad for?
You lose more water.
You're going to lose more water because the guard cells of stomata can't distinguish between water vapor as a gas and oxygen gas and CO2 gas. So you either have more effective gas movement or you don't. And there's no way to filter it out. There's no filter here that recognizes water vapor versus CO2 and oxygen. That would get incredibly complicated. So that will help you with the questions later on there about the pros and cons, advantages, disadvantages of some of these different types of leaves. Okay, here's a, happens to be a corn, and some monocots have this and some dicots do. So again, this is not a distinction among the types of plants. It's an adaptation. That some of these have at uniform intervals in the upper epidermis, these three cells. What are those called? Bulliform. And some of you were asking me this, and I noticed it wasn't in the lab, so I need to make sure we go over it, is the bulliform cells, it tells you in the lab, lose water quicker, which is going to lower the pressure. But why do they lose water quicker? Can you tell by looking at the slide, or the slide you were looking at? There's an anatomical reason that they're going to lose water quicker. The cuticle is thinner. There's a cuticle there, but it's actually thinner. So the red halo you see here and there, there's a thick cuticle on here that's so thick you can see it with the dyes. It's not actually red in real life, but they've added the dyes to it. And if you get here to the bulliform cells, it's thinner. So if there's plenty of moisture, all these cells have plenty of pressure. However, to help you with the questions in the lab manual, is that if you have a leaf, and you have bulliform cells on the upper epidermis, and especially if they're located right in the middle, when you get into a drought, you're going to have higher pressure below than above, and so the leaf is going to fold exactly in half. And that's what many plant leaves do. And now when it's folded in half, it's reduced its leaf surface area by 50%, less exposure to heat, sun, wind. And then a question in the lab said, well, some leaves don't fold in half, some leaves, the leaves curl. So I'll do something like this, and now the leaf surface area is not reduced by 50%, but probably 75 to 80%. That's a pretty good survival strategy, and they did this without muscles and a nervous system. What do you think is the difference in the anatomy of a leaf that curls versus folds? It's a fairly simple difference, and it makes all the difference. You get it?
It has less surface area, so it's going to absorb less water.
No, it has something to do with the bulliform cells. It still is bulliform cells.
So it doesn't lose water.
Yeah, if it folds in half, the bulliform cells are mostly in the middle. But what about if they're scattered across the whole surface? Then you're going to get this curving and cupping, and so this rolls up. This is the number of bulliform cells. And all this gets back to is that what the video alluded to, which is one of the main ways that plants move things rapidly. There's no muscles, there's no nervous tissue, there's changes in pressure. That's another advantage of having cell walls, is that they just can rapidly change pressure. In this case, it's rapid in the sense of it can happen in a few minutes. It can go from being totally flat out to curling out. That's a potential advantage in plants that live in areas prone to drought. So, looking at this, this particular corn plant has two features that really help it to reduce water loss, but it has a feature that enhances water loss, so that kind of balances out. It has a thick cuticle, so that helps reduce moisture loss. It has bulliform cells, which helps reduce moisture loss. But what increases moisture loss? Yep, guard cells upper and lower. So corn is in a balance. Corn actually tends to lose a lot of moisture. Corn, if you ever grown corn or all the acreage we have in this country in corn, it's hundreds of thousands, millions of acres. It takes an enormous amount of water. But when it has that water, it's very quick growing. It's very efficient at photosynthesis. And part of it is these guard cells upper and lower, but the other part that we'll get to in the photosynthesis chapter is there's something a little bit different about the spongy mesophyll and this vascular bundle. It's a different type of photosynthesis. C4 photosynthesis. And we'll get to that later. Okay? This is what you might see if you did a really good epidermal peel, meaning take a leaf, turn it upside down, peel off the lower epidermis only so you're not getting mesophyll cells, and there's usually no pigment in the epidermis except in the chloroplast. Except in the chloroplast of the guard cells. So here's a pair of guard cells, that are closed. You can see the chloroplast. And then, so a pair of chloroplasts, an opening there would be the stomata. And then these other cells, here's the nucleus. That's the parenchyma cells. Because remember, epidermis can have trichomes. It can have guard cells. It has parenchyma cells on there. So it shows you the dramatic difference in shape of a parenchyma cell to other epidermis versus guard cells. But you should realize that when we go back to this. Look at the difference in the guard cell shape there versus the normal parenchyma cell in the epidermis. So that's reflected in the cross section, but here it is on a surface mount. Quite a bit different. Here's a prepared slide that you had, and it shows that some epidermises of plants, this looks like a jigsaw puzzle. And studies with physicists have actually shown is that really helps to hold the leaf together. If you look, a leaf is really an engineering marvel. Because how does it stay flat out most of the time for photosynthesis? It has to be thin in order for light to go through, but it has to be strong. So part of it is the vascular bundles. Part of it is the interlocking epidermis, and you can really see that best here. And then, what in the world is this structure? Something we've had. Trichome. This is a trichome with a gland at the top, and that gland could be producing a pesticide, or it could be, if it was on a carnivorous plant, it could be like what the video is talking about. But that's something that's very sticky, that tends to keep insects nearby, and then it folds up, and too bad for the insect. Okay, this is a longitudinal view of a stem and a leaf, and you have a slide of this. And what's interesting, we'll be talking about in lecture, is that it has a preformed excision zone. So when the plant gets into a drought, or it's coming up soon, once we get to October, November, leaves start falling off many of the trees, it actually abscisses right there. This is all leaf tissue. This is stem tissue. This is a piece of what looks like a lateral bud. And then this abscission zone, by the time this leaf falls off, has already, some parenchyma cells there have already started to form a new dermal covering. And so then the plant loses the structure, but it's not losing water and not prone to infection. So that's, and the reason it can do that so quickly, leaves can drop quickly, they have a preformed abscission zone. Okay, this is now getting into what's in the back of the room with the Now we're getting into leaf specializations, and this is a little bit on the video. These would be the trigger hairs the video talked about.